Okay, all right.
Yeah.
Should I start?
Yeah, let's start. I think it's 3 o'clock.
All right. So thanks everyone for joining. This is my first webinar, very exciting. Thanks Eski for inviting me, and thanks to the ISCB RSG-Turkey for inviting me. Thanks everyone for joining. So I'm a group leader for population genetics here at the Max Planck Institute for Science of Human History in Jena, specifically in the Department for Archaeogenetics. And archaeogenetics is a rather new term. It's a bit of a made-up term that basically comes from merging together archaeology and genetics. The previous term was called or one wants to make clear that we're actually talking about specifically archaeological remains, so human remains, and often also in a little bit different time periods than, for example, paleontology. Anyway, archaeogenetics is the umbrella term here in the Institute, but I'm actually a population geneticist, so I'm very generally interested in how people are related to each other and what we can learn from that about human history. And as we will actually see, somewhat ironically, the deepest results that we so far have about human history actually come from analyzing modern DNA. They are not necessarily based on ancient DNA, but of course, ancient DNA will still continue to give lots of new results on the more recent past, and I will show several of these as well. So let's start. So a bit of an outline for this talk is I'm going to give 3 sort of examples on what we're doing. The first one is investigating deep human population structure with MSMC and MSMC-IM. So that's quite recent development that I'm currently doing with one of my PhD students. That's all based on modern DNA pretty much. And then I'm going to give 2 examples of recent ancient DNA projects. But actually, the most recent published one is on Fennoscandian genomes. Maybe you've seen that. That's a paper that just came out last week in Nature Communications, but I'm not going to talk about that today since I realized I didn't announce it properly in the abstract. And anyway, these 2 other projects are also exciting. I guess there's always more to talk about than one can. But anyway, for this seminar or webinar, I chose 2 ancient DNA projects then. So this Anglo Saxon migration project in Britain, which we finished a while ago, but which we're actively working on now again. And a project where Eski actually is working with me and other people from Ostrava and Czech Republic on reconstructing human population history in North America using some new methods. Okay, so someone says, I can't hear. I hope that's the only one, because I haven't heard any other complaints from someone else. But anyway, I'm going to continue. I hope that someone will shout if more people can't hear or something's wrong. Okay, let's continue. So investigating deep human population structure with MSMC and MSMC-IM. So that's the first part of this webinar. And maybe I should start pointing out that whereas human genetics has now described human genetic diversity across the entire planet. Lots of people have been sequenced. Lots of ethnic minorities have been described genetically. And there's sort of 2 angles to how to go about this. One is to just describe genetic diversity and in particular analyze its connection to disease risk and other phenotypic variation of human global diversity. But there's another way to look at this, and this is for trying to understand how people are actually related to each other. And that is shown in this next slide, where instead of trying to understand specifics in these people's genomes, and as I said, like which genes are responsible for which genotype or so, we're trying to understand how these genomes, what these genomes can tell us about a time scale and a possible and a topology or a pattern of how human genetic diversification came about, and what that tells us about historical events such as population splits and perhaps movements across continents. And I will not go into much detail about this tree on this slide, but I will derive it a little bit later on what it all means and how we came to that conclusion. So in general, the process of learning about history from genome sequences works in 3 steps. So the first one is you start with genome sequences, obviously, and you identify genetic variation in them. And then you try to reconstruct genealogical trees. And this should be familiar for people who have worked with mitochondrial DNA and Y chromosomes, where there is exactly one genealogical tree that you can form for the entire molecule of one mitochondrion or Y chromosome. And this reconstruction works by trying to fit a tree to this pattern of mutations by looking at which genome is how distant to which other genome, and how can that all be described in such a genealogical trees, connecting all samples that you're looking at into a tree to the most recent common ancestor of all. So that would be mitochondrial Eve up here, for example, if those were all mitochondria that we're studying. Someone asked about that. I may talk more about this in a bit. And from there, actually, we're trying to understand what is the demographic process behind this that shaped this tree. So the tree alone yet doesn't tell us anything about migration or whatnot, because the tree itself is just a random sample, or it contains lots of randomness that then has to be that then leads to a statistical inference of the underlying demographic processes, such as population size changes in time, or population structure and population splits. And both of these processes are connected with high uncertainty. That's what makes it statistically interesting and very challenging. So in particular, well, curse and a good thing both at the same time is genetic recombination, which describes that if you look at other things other than mitochondrial DNA and Y chromosomes, autosomal DNA, we actually inherit, of course, 2 chromosomes from each parent. And there's going to be a mosaic of all of our ancestors in our genomes. And this makes it even more challenging, but also richer to analyze, because now we realize that we haven't only one tree that connects our entire genome, like is the case for mitochondria or Y chromosomes, but we have thousands of trees along the sequence. And this makes it very challenging, because not only do you not know where one tree ends and the next one starts, so you have now multiple trees across these sequences, and they are separated by ancestral recombinations, but you don't know where they start and end. And even if you knew that, you don't know exactly how to reconstruct these genealogical trees from the rather few genetic mutations that are sprinkled on the sequences. So recombination rate and mutation rate are about the same scale, so this makes it a very interesting statistical problem. So some progress was made using methods such as the SMC, which is the sequentially Markovian coalescent model that was published in 2005, 2006, and then people built inference methods on that. I was rather late to the game. This is one of these processes, and there's some citations down there that I don't want to go too much into detail, but that explains how you can derive a generative model that explains how one tree changes to the next tree. We used that in 2014 in our model MSMC, but actually this was just a successor to another model called PSMC by Lee and Derb in 2011, which basically used this process of how trees change along the sequence to statistically sum up all possible paths, all possible sequences of times to the most recent common ancestor of a single pair of sequences. So now we gave up the idea of deriving entire trees. We basically went back to the idea of having a single diploid genome, which means one chromosome from your father, one from your mother, and we're trying to reconstruct the sequence of times for the most recent common ancestor along the sequence. And that was extremely effective and allowed Lee and Durbin to derive relatively high resolution demographic history curves for individual persons, which was something unheard of at that time. We drove this a little bit further and extended it to multiple sequences. Let me check whether, okay, I should explain this. This plot actually shows that you can actually reconstruct a sequence of times to the most recent common ancestor along the sequence. So this just shows a chunk of the chromosome, and where the black curve is very low, you have a very recent common ancestor, like in the circled one here, and in other areas you have more ancient times to the most recent common ancestor going back millions of years. And you can reconstruct that probabilistically, as shown here in this heat map, where we can really use this method to efficiently get a probability distribution of times to the most recent common ancestor across the sequence. And from there, we can infer high coverage, high resolution demographic history curves, so population size curves. I'm jumping over lots of machinery here because I want to cover some ground, but going from there to the demographic history is not trivial, but I anyway want to jump there right now and show what we can do. So this is now a result from MSMC 2, which was derived from one of my students, K. Wang, who ran this on a number of simulations that we did. So here's a rather ridiculous simulation of a population size going up and down by a factor 10 over millions of years. So you see to the right of this plot, you see millions of years, and to the left you see a couple of thousands of years, and you see that population size shoots up from about a factor 10 every so and so many years. So a rather unrealistic scenario, and we're trying to basically infer that from single genomes. So here's the curve that was obtained from a single genome using our methodology, and you see that it kind of captures well genetic effective population size between a couple of millions and a couple of tens of thousands of years ago. And then the thing we added to the game is that you can now add multiple genomes and increase resolution towards more recent time. For example, here up to 8 haplotypes. Now you can infer population size history up to 2,000 years ago. Okay, so that's all published in 2014. Nothing's too new about it. I want to now show some results from real data. This has been published in 2016. Very nice paper by Malik et al. This shows the SGDP panel, the Simon's Genome Diversity Panel, which released 300 high coverage whole genome sequences from the entire world. Lots of them from interesting ethnic minorities and interesting populations that are unadmixed of colonial or more recent processes. So this is a very valuable resource for us. And we ran it on a couple of genomes. And this is what we found. So this is our kind of first glimpse into human history from real data, if you will, in this talk. So here we've analyzed the population size curves using this methodology, MSMC, from a dozen or more pairs of individuals from a single population. So, for example, 2 individuals from the San, 2 individuals from Buti, these are Central African rainforest hunter-gatherers, 2 individuals from Dinka, an East African population, and so on. So I'll tell more about these populations in a second. I just want to highlight some of the results that we found. So on all of these plots, the distant path is on the right, so here, and the recent path is on the left here. And you can see that in all of these non-African populations before 50,000 years ago, so all these non-reddish and yellowish samples that are circled here, we see an identical population history that diverges from their African cousins around 200,000 years ago, and then goes all the way down to a steep bottleneck around 50 to 60,000 years ago, which archaeologists can connect to the out of Africa dispersal event. So the first anatomical modern, or actually the first main dispersal out of Africa in recent times of anatomically modern humans. There are some traces of anatomically modern humans also after that. So I just got a question here. Any individual from Turkey? No, unfortunately not. I'm not aware of any high coverage modern Turkish genome, but I should probably check. There are. SG tells me there are, so maybe we should run it on some Turkish genomes. We should have run it for this webinar on Turkish genomes. Anyway, I suspect they look not too different from other Eurasian genomes that we have. So then anyway, then you get this kind of two, because I should say that's not because there's nothing special about, that's not because the population history is all the same in all of Eurasia, but the resolution of this method is not too fine to kind of look at these extremely detailed population diversification processes in the Holocene. This is more about deeper population history. Okay, so what else do we see? We see, I need to rush up a little bit. So we see that in African ancestors, we see a more mild bottleneck that is not comparable to this deep bottleneck seen in the in non-Africans. And then a little bit moving forward in time, we see that in Native American populations, we get an even deeper bottleneck after that. Around 15,000 years ago, it has this deepest point, and it then shoots up. And that's also very nicely connected to an archaeological event, namely the first evidence of anatomically modern humans in any humans, I think, in America. So when Native American ancestors split from their Asian cousins and crossed the Bering Strait and entered North America, this was about 15,000 years ago. And we'll hear more about that actually in the third project I'm going to present. Okay, so then there's more complex patterns in the Holocene. We've looked into more detail in that as well, but I can't go into these details now. But there's lots of rich stories to tell from these plots. And I now want to move on and now say what we can actually do about cross-population analysis. So all that was now based on analyzing a single population at a time with no interaction. But of course, this framework goes deeper than that. We can also look at how populations are related. And we do that with analyzing these three aspects of population gene genealogies. Now, let's assume we have sampled genomes from two populations at a time. Then we can ask, what is the average time to a most recent common ancestor, or the distribution of time to the most recent common ancestor between haplotypes within each population and also across. So if two populations have been isolated for the last 40, 50,000 years, then, for example, you would not expect many genes to coalesce, so to have a recent common ancestor across these populations that is younger than 50,000 years. And we kind of munched all that up and created a statistics which we call relative cross-coalescence rate, which is a simple statistic that crosses over from 0 to 1 as you look back in time. Okay, so this, for example, is again a simulated case where we simulated a population split 100,000 years ago. So two isolated populations that are isolated since 100,000 years. And as you look back in time, this statistics crosses up all the way from 0 to 1, indicating that around that time, the split of the two populations occurred. And indeed, this simulation shows that it works. Indeed, this crossover from 0 to 1 happens at about 100,000 years ago, the exact time which we simulated. So if we now apply that to real data, we see a nice grading of different separations in time, starting with the earliest ones from Africa. So these reddish curves here are separations between the West African people, the Yoruba from Nigeria, from lots of non-African people, Americans, Asians, Europeans, and they all fall exactly the same shape and fall around the same time, about with a midpoint about 70,000 years ago, but going all the way back to 200,000 years. And then you see so these are these here, and then you see other splits between Asia and America, and between Asia and Europe here in bluish, and between Asia and America here in greenish, and other more recent events. So this is the point where we now kind of did something new. So up to that, this is a summary of what was published 2014 and 2016, the newer version of the method, but basically this is old stuff. What we now added is something new that I now want to talk about. So basically, we've reconstructed the human family tree using these kind of pairwise estimates and pairwise analyses between Africa, America, Asia, and Europe. And here's a summary of that in time. It's kind of complicated, and there's admixtures between, as you see with these stripes, so some of these things are not very sharp. Anyway, what we now did is look a little bit deeper into time even. So one question we asked is that really all to it? Is really all of human history diversifying in the last 200,000 years, or is there more to it? We already know that there is more to it, because we know of Neanderthal divergence and Denisova ancestry, so archaic human genomes that contributed to different populations in different amounts. So we know that this is not all of the story, but there are streams of ancestry that go back deeper. But is there more to it? So we developed a new model to look at these kind of cross-coalescence analysis in a little bit more detail. This was done by my student Kei Wang, who derived an isolation migration model for the connoisseurs. Isolation migration is kind of is a well-known parameterized model in population genetics. Here we added some flavor to it. We actually added the fact that migration rates are now changing through time. So we have two isolated populations. So we act as if we analyze two different populations, both of them live on an island, and they can exchange migrants over time as you go back, and the migration rate changes through time. And now we want to infer that change in migration rate through time. So as we now test this, and again, I'm jumping over lots of machinery, we can now analyze splits between two populations more quantitatively. So here on the upper plot, called migration density, we now can infer during which point in the history these two genomes exchanged migrants and when not. So in other words, when was the bulk of this kind of coming together of the two populations as you go back in time, and when were more periods of isolation. And this is a little bit hard to understand if you look at it the first time. So let me try to basically go through some real results, which may help you to give a little bit of intuition for this. So here are some simulations where we have a clean split. So with a clean split, we simply have two populations that split at one point in time. And sure enough, in our simulations, we see that around the split time, the simulated split time here going all the way from 5,000 years all the way down to 300,000 years, we see one peak of this migration profile, indicating, as we expect, that around that time of the split, the two populations come together and exchange lots of migrants, and that reflects their split time. Okay, so we now have a way of parameterizing a clean split using this kind of migration profile. But now we can do more complex things as well. So for example, we can ask, what do we see if we have a split and post-split admixture? Okay, so here between 10 and 15,000 years ago, you have an admixture period. So now we should see two peaks. And sure enough, that's what we see if the split time and the migration phase is separated well enough. So for about 50,000 years split time and 75,000 years split time, we actually start seeing two peaks in this migration profile, indicating that the bulk, that some part, actually 50% of all lineages, are moved together as you look back in time around this migration period of 10 to 15,000 years ago, and the other half remains isolated. So that's why you see a dip in the profile again, and then you see a second peak around the time when the rest of the genome merges together. Okay, and the third scenario we simulated is one with archaic admixture. So here you have a split time between POP1 and POP2, and then you also have in one of the two populations a contribution from an archaic population that split much deeper in time. And again, this is also something where you should see two peaks, one peak around the time of the split between POP1 and POP2, but that shouldn't bring all the ancestry together, because parts of that ancestry should go back all the way to Neanderthals. For example, if you analyze Africans and Europeans, then indeed you should find that about 98% of all ancestry should probably come together with Africans around 100 to 200,000 years, but 2% should go back all the way to 400, 600,000 years, or even later when Neanderthals and Africans lineages finally emerge. So that's what we see, and now let's look at some real results. And there was a big surprise here for us when we analyzed African pairs, so pairs of African sequences. This is all within Africa pairs. We found that this migration profile extends much deeper than we thought. So from all the analysis before, we thought that basically human global diversification is more or less finished in Africa around 200, 300,000 years ago. Now we find that the signal of deep ancestry goes much deeper than we thought, potentially even 1 million years and older. And that was a surprise to us, because that wasn't really reported before, at least not for San and Mbuti, which are the two populations. These are two hunter-gatherer populations from Africa, one from South Africa and one from Central Africa, for which there has been no reports of archaic ancestry yet. But here it seemed to suggest that there is archaic ancestry in some of them. That basically means that a tiny, tiny proportion of ancestry, it's almost only 1% or less, I will show this more quantitatively later, doesn't converge at 300,000 years ago, but goes all the way back to 1 million and older. So that's a new result. We also found some other things. For example, African, non-African pairs have this two-peak distribution, which matches our expectations in some way. So, for example, Yoruba Australian, but also Yoruba French, Yoruba Sardinian. These are pairs of African, non-African populations, where you expect that most of the ancestry comes together about, midpoint 70,000 years, but going up to 200,000 years. But then there is a fraction that goes through archaic ancestry and is older. And that's what you see here. The timing of this second peak is not quite correct. If you think it should be archaic ancestry, we know that these estimates between Neanderthal and modern human divergence should be more 600, 700,000 years ago. But we have seen from simulations that this second peak gets kind of moved and squeezed a little bit more to the rest if you don't have much of that signal. And here, of course, we're talking about small signals, so we were surprised to actually see anything. But there does seem to be a systematic second peak in most of these pairs of African non-Africans. Okay, so I think now I just want to summarize that bit. So this kind of deep ancestry can be visualized here. So this is basically the proportion of ancestry in these pairs of genomes that is older than 1 million years ago. And you can see that, for example, between San and Dinka, or San and French, but also San and Sardinian, San and Yoruba. So pairs of populations involving Africans, in particular involving San and Mbuti here, a little bit more to the right. So San and Mbuti, you get these residuals of order 1%, 0.5% of extremely deep ancestry. You don't see that in most of the rest of the populations here. Reassuringly, of course, we wouldn't expect that for many of the pairs, where we know that the history is not that old. A little bit of a different view to look at this is this, where we say 99% of the ancestry is actually younger than, and then we give the number. So for lots of these San and Muti pairs, we get 1 million years and 700,000 years and 600,000 years. And for most of the other genomes, we are much more in the bulk of diversification time that has been reported before. So about 300,000 years where human diversification starts. Okay, so that was a lot to cover on that front. So you saw that deep human history can be recovered from modern genome sequences, somewhat ironically, because of course old DNA, one would think, tells us more about old times, but that's not always true. So here we're really exploiting rich information from the process of how DNA gets inherited through mutation and recombination. So conclusions, yes, I said that genome sequences provide rich information about our past. These methods like MSMC can model that efficiently, and we can now learn about these demographic history, but also population structure with more details, and potentially reveal some signals up to 1 million years ago, which is arguably new. Okay, let's move on to the next topic. So the next topic is now the opposite end of the timeline. So we've talked about millions and hundreds of thousands of years. Now we're going all the way to the most recent events in human history, where we actually already have historical evidence and linguistic evidence, and that is the early medieval time in Great Britain, where we have the Anglo Saxon migrations. Okay, let me just check some questions here. Have you tried this new method to estimate migration rates between Neanderthal populations? No, we haven't. We have, though, applied this method to pairs of Neanderthal, non-Neanderthal sequences for some of these high-coverage Neanderthals. The one thing that I'm a little bit afraid of when applying this to archaic genome sequences is that they need to be phased. This is one of the details I completely skipped over. Phasing means that you have to resolve paternal and maternal haplotypes. And that usually works with a statistical process that relies on thousands or hundreds or at least dozens of other genome sequences from the same population that isn't yet available for Neanderthal. So at the moment, phasing of Neanderthal genomes is dodgy and not so reliable. And that's why I'm kind of a little bit afraid of interpreting too much into this. Some of the results that we have seen so far from both Denisovan and Neanderthal pairs with modern humans in terms of divergence time make sense and are consistent with previous results. So there's no surprises there, but we haven't investigated this deeply. Okay, are there more questions about this part? Otherwise, numerical artifacts and unexpected peaks. Did I write this somewhere? African, non-African splits.
Could you please read the question?
Yes.
Because in the record.
Oh, yeah, I see. Yeah. So the question is, what did you mean by numerical artifacts and unexpected peaks for the African, non-African splits? What did I write there? Let me just check. Denisovan Neanderthal integration too young. Yeah, that's this thing that I meant that we saw in the simulations, that even if you simulate a Neanderthal or Denisovan integration, that came even if you simulate the divergence time between Neanderthals and modern humans of 600,000 years ago, you don't really see that in the profile. Here's an example in the simulations. So, for example, I'm circling this one and this one here, where you see that you should actually see two peaks cleanly at the two lines, which are the lines corresponding to the split time between POP1 and POP2 and the split time of the archaic population. But you actually see some tendency that the two peaks appear closer together than they should. So MSMC is not able to actually resolve these two processes, these two peaks completely independently and completely separated. But there's some tendency to move them together. So this time estimates of the second peak or third peaks cannot be fully trusted. But the tendency is that it's too young. So I am confident that the signal we see about 1 million years ago looks like an archaic signal that is very old. Okay. Let's move on. I'm happy to answer more questions about this part also later, because people need to write them, of course. Okay, let's talk about Anglo Saxon migration. So probably most of you know that in the early medieval time in Great Britain, there was, at least historians tell us, and archaeologists tell us, and the language change also tell us, that there was a large migration from the continent, arguably from Germanic-speaking areas like Saxons and Jutes and Angles. So these are people from the continent in Denmark and Holland and in Germany, Niedersachsen, for example, in Germany. And these people then left their home and went into Britain and settled there. And we don't know much about this process. The only thing we know for sure is that it changed the language drastically. So before that, arguably all of Britain was probably Celtic-speaking, everybody in Britain spoke Celtic languages of one sort or the other. So of course there's some differences, but certainly not Germanic languages. And then it changed. And since then we have modern English, which is, or actually Old English and so on. So the English language itself, which is primarily a Germanic language. So it did change that, and it did have other cultural impacts as well, as archaeologists tell us, but we don't know too much about the process. So we actually looked at this, and two papers were published in 2015 about this. One from my former group where I did my postdoc here, the upper paper, and another paper from Martignano and colleagues. And both of them released together the first British DNA, ancient DNA from ancient archaeological remains from Britain. So, and I now can give you a meta-analysis of these two papers. So that's a nice thing. Now we have more genomes than before. We now have about 20 genomes. So 10 samples from 3 sites close to Cambridge, around here, and then some more samples from sites close to York. That was the other paper. And in total, these samples span 4 time periods: the Iron Age in blue here, the Romano-British period, so Roman period, early Anglo-Saxon period, and middle Anglo-Saxon period. So what do we find when we look at these genomes? First of all, some statistics. So we sequenced those genomes. Both the teams, actually our team and the Martignano et al. Team, sequenced them shotgun. That means that we didn't create any local enrichment or something for genomic DNA, but we actually sequenced everything that we got. So we reconstructed full human genomes. Here I highlight you to this column here, which gives the genomic coverage, which is the average times that you cover a certain genomic position with an ancient read. And you see that for most of these British samples, for these Romano-British samples, it's about 1x. That was the other study. We had a little bit higher coverage, going up to all the way up to 12x. So very nice data actually. Okay, so what did we find? The first thing that we often do is in this field is principal components analysis. So this is a very common method by now. I will anyway introduce it briefly. So what it tries to do is it puts lots of genomic data into a 2-dimensional plane. So of course, every genome here, this is from modern people, from present day people from these different countries. Actually, Turkey should be there. There you go. Turkey is here in little stars, falling all the way scattered between these 2 lines in these brownish circles. That's Turkey. And every individual is of course, every individual's genome has about 600,000 genomic markers in this case, because this is an array typing, was done on array typing technology. And now we reduce this dimension of 600,000 to 2 dimensions and try to basically recapture their genetic distances to each other. So samples that are far away on this map are also more distantly related, and samples close to each other are more closely related to each other. And you see more or less 2 clines. You see a cline between Northern Europe and Southern Europe here on the left side, capturing most of Central and Western and Southern Europe diversity. And you see a different cline here all the way from the Caucasus down to the Levant, to the Saudi Arabian Peninsula or Arabian Peninsula. And in between this, you have some bridges spanned by, for example, Turkish, but also Greek and other Balkan people. Okay, if we actually, now let me clear this. Now, if you actually map the modern genomes, the ancient genomes on that map, you get a little bit of a surprise. So first of all, you see that most of these samples now cluster left here on the left-hand side of the plot, close towards modern British and modern French and other European people. One individual falls all the way back to actually close to Syria, close to present-day Palestinians and other populations, indicating that this guy actually was a soldier. So this is a Romano-British person from York. So this soldier was arguably someone from that area who was recruited into the Roman army. So that's not too surprising, but it's still kind of cool to see that the Roman Empire was actually genetically as cosmopolitan as it politically was. Okay, so let's look and zoom in a little bit into this blob of European samples that we got from this study. So you see that they all fall close to modern day English and French, but there isn't too much of stratification between them. There isn't a clear pattern where you could say, aha, Anglo Saxons look like this and Iron Age people look like this. It looks more like a cline between the two. And arguably at the time, we believe this is partly also due to resolution of the method. So principal components analysis isn't of course a method that can solve everything. So we hoped to find something else with different methods. And what we turned to at the time was rare genetic variation. So rare genetic variation was shown in other projects to reveal more fine-scale population structure, like the 1000 Genomes project. And indeed, what we found here is we found when we did a very simple counting exercise where we identified singletons, here marked in brown and green and so on, in these reference populations. So here Dutch, Spanish, Italian, and Finnish. And we now ask for a given test genome, what is the rate of sharing of mutations that are, of course, here highly exaggerated? How many of these singletons do you share? Does your genome share? You can actually get a fingerprint here, a profile of how much you share with different populations, and you can use that to distinguish samples from each other. So for example, when you do that for Iron Age versus Middle Saxon samples from a site close to Cambridge, from Hingston, actually this is the site where I did my postdoc, coincidentally, you find two different profiles. You find that the Iron Age samples, who are 400, 600 years older, I'm sorry, more ancient, have a different profile, where in particular sharing between, for example, Spanish here, IBS, and Netherlands is more on the same level as in the Middle Anglo-Saxon samples, where there's a lot more Dutch sharing, right, if you look at these three profiles. So now you see clear differences between these groups. And we could condense this down to a more useful summary statistic, perhaps. Here's this plot where we now have all the individuals, the ancient individuals along the x-axis, and we record how much Dutch sharing, how much sharing, allele sharing of with rare alleles ascertained in present-day Dutch do they share relative to alleles shared with present-day Spanish. You see that these two levels that I also highlighted in the previous slide, so you see the Iron Age population here down at some lower level, and Middle Anglo-Saxons from Hingston and from York, I should say. This was very satisfying for me to find out that the York Middle Saxon sample that they had falls exactly in the same range. These two kind of form the two poles, and now we can look what do the other samples fall into. So first of all, we see some early Saxon samples from Oakington falling somewhat in between these two ends. So we see some that have high Middle Saxon ancestry, some that have these low Iron Age or Celtic ancestry. Then we see the Roman soldiers from York, which are also more on the Iron Age level actually overall, except for this guy, which was not reported by Martiniano et al., which seems to have some Anglo-Saxon ancestry. So at least on this statistic. So that's kind of cool. And now we can ask, where do the modern British population fall? And they actually fall somewhere in between. You see, now have a level around here and a level around here, and the modern British fall at a level around here. And you can, of course, do this more quantitatively as in the next plot, where we have these mean levels, and you see that there is somewhat like an I would call it an immigrant or a foreign genetic signature indicating immigrant ancestry in the Middle Saxon and early Anglo-Saxon population, and an indigenous gene pool reflecting more the Iron Age level of the Iron Age gene pool here in blue, and the middle and the modern English or present-day English are somewhat between. So that gives you kind of a detailed view on what might have happened in the Anglo-Saxon migrations. Of course, that's only 4 or 5 sites, but I should say that we now have 75 more samples in the lab across all of Britain. So we're looking at this question much more detailed. But what we can already say from this are 2 things. First, there was some heterogeneity in early Anglo-Saxon times, where certainly in Oakington here in orange, we see that some fall low and have this Iron Age profile, and some have this new immigrant profile, and with some admixture between. And those people were buried on the same graveyard, so it means that the mixture must have happened in the communities. It's not the case that there were communities which looked entirely like this genetically or entirely like this, but there were mixed communities as well. So that's kind of cool. That's one thing we can say from this. And the other thing we can say is that present-day English people seem to be at least consistent with a mixture between these 2 gene pools. So you have the Iron Age and the Anglo Saxon sheet pool, and with modern present day English falling roughly between. More precisely, actually, we can model this and conclude that they are 38% Anglo Saxon if we believe this statistic. Right. Okay. So let me summarize briefly. Oops. Oh goodness, this is the same conclusion slide as before. I'm very sorry. Let's look at only the two lower conclusion points. So rare lead sharing provides a simple and powerful way to analyze subtle population structure and admixture, and it shows that the modern English population is admixed. So those are the two main conclusions. So some questions. Wondering what % of genomes were intact. Well, actually, all of them. I mean, at least all of. I see. You mean in contrast to what we tested. Well, for the Oakington site, for example, the early Anglo-Saxon site, where we only have 4 producing genomes, we tested, I think, 16 genomes or so. I should say that a lot more were successful from nowadays perspective. With our new technologies in the lab, we can now do this capture enrichment technology, which allows much cheaper sequencing. But at the time when I did my postdoc, we did this shotgun genome sequencing, which means that you have to sequence everything. So anything below 10% endogenous DNA would be prohibitively expensive to sequence. So we selected only the very best ones to sequence entirely for shotgun data. Nowadays, we would select everything that has more than 1% endogenous human DNA and then apply enrichment capture to look at least a set of SNPs. So this has changed a bit. But at the time, I mean, I should say the preservation in England is absolutely spectacular. It's phenomenal. We have many samples there that routinely get up to 50%, 60% of endogenous DNA. So there's something in the soil there that preserves DNA very well. Right. Okay. Let's kind of move on. So there's a last project I want to talk about. This is this project about North America. I'm now jumping back a little bit in time. Sorry for this order of things, but this kind of reflects the historical order in which I did these projects and in which these talks grew. I should maybe arrange it in a different way. But anyway, this is the most recent project that we did. That's why I kind of put it in the end. And it's a project where we analyzed in a little bit more detail and what happened in more recent times in North America. I already talked about in the MSMC project that North America or that the Americas in general were settled from a population that split from Asian ancestors perhaps 15 to 20,000 years ago and then moved into all of America. But it turns out that that's actually not the only movement that happened in America. So this certainly describes this so-called first people Pleistocene movement, 15,000, 13,000 years, moved into America and settled very rapidly all of America. There was a recent paper actually about South American genomics, several papers now that describe this process in more detail. Then there was a second movement of Paleo-Eskimos, so a population that archaeologists know that covered the Arctic for many thousands of years, from 4,500 years ago all the way up until 700 years ago or so, when they were finally replaced and went extinct as a culture, at least, by Yupik and Inuit, who are representatives of this last migration that happened only 1,000 years ago, the so-called Thule culture, which is the name of the archaeological culture. So, of course, we're not the first ones to look into this. There's many previous papers about this. There's one from David Reich and colleagues from 2012. There's one from Manasa Raghavan and colleagues in 2015, and a more recent one from Victor Moreno Maya et al. From this year. And I just want to I don't want you to pay much attention to these models, which are very complicated and of course deserve their own talk. But I want to highlight that there's one population called the Athabascans, which shows up in all 3 of these models. Here, Chipewans, this is a subgroup of Athabascans, and also here, which play a special role and appear admixed in each single one of these studies. And that was one of our leverage points that we wanted to look at to understand better how are they admixed and what does that tell us about these different movements. So we collaborated with David Reich's team in Harvard about this, and in the end were extremely fortunate to be able to work on 48 new ancient genomes from the American Arctic. So they fall in different groups. There's 3 ancient Athabascans from Alaska, a sample from the Canadian Arctic, several Aleutian Islanders, and several ancestors of modern Yupik and Inuit, so modern Eskimo movements from Chukotka, and some people from more western sites. They're all kind of displayed here in stars. I'm not going to go much into detail on where exactly they are, because this plot also shows reference populations, and it would take me too long to highlight them. But anyway, so these are the 48 ancient populations, and the first thing we looked at was a principal components plot as before. So now it looks a bit different. So here we have West Eurasians here on the left. We have South Americans here on the upper side, and we have Siberians and Chukotko-Kamchatkans, or actually we have Siberian populations here on this lower triangle corner. And you can see that all of our new ancient samples that we kind of published, except one, and I'm not going to talk about it, by the way, fall onto a cline between this kind of Siberian people and South Americans. And this cline is, of course, something you would not expect if there was only one movement. Well, it's not surprising. We already know that there's not one movement into America, but there seems to be different movements into America, also genetically, that contribute to different degrees to these different populations. With kind of the most extreme end on one end here, these are Paleo-Eskimos. This is the Sakak, the famous Sakak genome from Greenland, 4,000 years old. And then moving over modern Chukchi people to modern Yupik and Inuit people from North America and the Arctic, all the way down to modern Athabascans and then South Americans. So there's this kind of cline, and we wanted to test how that came about. And I'm going to jump now over lots of analysis that we did for this paper. You can maybe read some of it in a quite outdated preprint that we now still have on the web from last year. It's going to be massively updated soon, hopefully. And so one of the new things we brought to this project, I think, was that we employed these new methods that we came up with for the Anglo Saxon project based on rare variation. So here's a plot that Eski helped making, actually, with one of my students, where we looked at rare allele sharing between every of these newly reported ancient samples and some modern populations from America with populations outside of America. So with Chukotko-Kamchatkans here on the x-axis and Siberians on the y-axis. And you can see that there's actually two clines. There's one cline here going all the way from Southern America all the way up to Sakak, which is the Paleo-Eskimo. And you can see that Aleutes, ancient Aleutes, as well as ancient Athabascans and modern Athabascans fall nicely in between them, proving that they are actually consistent with a single admixture between Paleo-Eskimos and Southern Native Americans. In contrast, Eskimos fall outside of this cline. They are more shifted towards Chukotko-Kamchatkan ancestry, which suggests that they inherited something extra on top of this simple two-way admixture model from the continent. And that's indeed what we also find when we do modeling. So we can compare this, by the way, with common allele sharing. So this is based on rare allele sharing. So this, just to highlight what these kind of new methodology brings and what the power is that we add to it, by looking at these rare variants, we have these very neatly defined two clusters, for example, between Southern Americans and Athabascans with relatively small error bars. In contrast, if you look at if you look at total allele sharing, so including all variants of all allele frequencies, including common allele frequencies, you see a much more blurry picture where Athabascans kind of morph a little bit into the South American gene pool and a little bit of a less clear pattern also with respect to all these populations with larger error bars, at least relative to their position. So that shows a little bit that there is indeed power for this question to look at rare genetic variation. And indeed, we did more on that. So we actually also did some modeling, and this is a slide that kind of shows a little bit of the methodology we developed there. This is also a method that I developed for the Anglo Saxon project, but I didn't really introduce it there because at the time it was really used rather superficially. Here we now use it really heavily. It's called RareCall, and it's a method that not only tries to basically come up with clever summary statistics for rare allele sharing, but that actually tries to model rare allele sharing in context of an explicit demographic model of split times and population sizes. And the way it works, very briefly, I don't want you to understand all the math now in half a minute, but just very briefly, the way it works is that it exploits the fact that if you see a rare variant shared here between population 1 and population 2 in that manner, you know that these three variant sites must come from a common ancestor and that the mutation on that common ancestor that gave rise to these rare variants must have happened on the common ancestral lineage that connects all three of these variants. And that gives you some mathematical probability model to calculate what is the probability to see certain allele sharing across populations under some models. So a very simple result, for example, to give you a little bit, yes, this was capture data. This is one of the questions. Yes, the American data was capture data, except for 3 individuals which we sequenced shotgun. So that's an important thing to highlight because we wanted to use this rare variant analysis, and rare variant analysis works only on shotgun data. So the genomes that I showed here on this plot are all shotgun genomes. So an ancient Aleut, an ancient Athabascan, and this Sakark individual are all shotgun genomes. So as to give you very briefly some intuition for how this works, if you have more isolation time between 2 populations, so if you're talking about 2 populations which are more distantly related to each other, for example, West Africans and Europeans today, you would expect a lot fewer rare variants shared between these 2 populations than when you're looking at 2 more closely related populations, for example, French and Germans or so. So the number of rare variation shared between 2 populations tells you something about their distant relatedness. And to make that quantifiable and to actually calculate this exactly, we can use this RARCOL method, which does a kind of a clever approximation to it that makes this whole fast and that lets you infer in whole population trees. I'm going to show one of the examples. So, okay, this is a simulation try. I think I'm going to skip that in the interest of time. Actually, this just shows that it works. We simulated a population tree and we inferred a population tree from it, and it looks very similar, and the parameters are inferred correctly. I'm not going to go much into more detail, but I now want to jump to the actual results that we got from the data we analyzed here. So here we analyzed lots of modern European whole genome sequences from across the world, including actually at this point, not yet including the ancient genomes that we have in this project. This is also based on modern populations. And this was difficult to derive and took me a long time, but in the end, we have something that makes sense. Also, given other methods, this makes sense. So we infer a global population split between Europeans and Asians here with some error bar that I'm not showing around 40,000 years ago. This is slightly too young, which might have something to do with how the method works, but I'm not so worried about this because RARCOL is really more about recent events than about these deep events. There may be some biases towards more recent underestimations, about underestimations of old split times. But anyway, all the other times actually make a lot of sense. So you first see a split between Northern Asian or Siberians and Southern Asians, here Southeast Asians, around 22,000 years ago. Let me clear this out. Then a split between Siberians and Native Americans around 20,000 years. That also makes a lot of sense genetically and is in line with what other people have reported. And then you see splits between Northern and Southern Native Americans, and then later between Siberians and Chukotko-Kamchatkans and this more recent Eskimo-Thule expansion. So I'm very pleased by these split times because they match extremely well what archaeologists would also say about this, and I think it gives me some confidence that we're doing the right thing here. Of course, of utmost interest is, of course, these admixture rates here that we find. For example, we find a 10 to 15% gene flow between what we call Paleo-Eskimos here and Athabascans, which I will now go more into detail. We can now look into this this shaded area here in the zoom in a little bit more carefully. So we see that Chukotko-Kamchatkans here split from Aleuts and Yupik Inuit around 5,000 years ago, and then gave rise to 3 things. First of all, they contributed to Athabascans, which is 11 to 15%, which is important because that was one of the disputed points on where Athabascans got their genomes from. Well, we think that it's from Paleo-Eskimos. Before then, Paleo-Eskimos mixed with South Americans, or with northern First People, but non-Athabascans, giving rise to this mixed population that then gave rise to the Thule expansion. So everyone belonging to this Thule radiation, so Aleuts, Yupik, Inuit, have 50% Native American ancestry and 50% Asian ancestry. So it makes sense to assume that this gene flow event or this mixture event between Native Americans and the Siberian side happened on the common ancestor of Aleuts, Yupik, and Inuits, and that's indeed what we see. Then very intriguingly, we find a more recent mixture event. This was actually detected by Pavel Flegontov, ESGI's supervisor, with another method that now we integrated into this model and makes a huge amount of sense. So then there was additional gene flow between Yupik and Inuit and Chukotko-Kamchatkans, which finally explains why in Chukotko-Kamchatkans we find some residual Native American ancestry. So that was brought into by admixture across the Bering Strait between people living on both ends of the Bering Strait. So that was quite pleasing, and a later addition to the model that is not yet, for example, in the version that we have on the on bioRxiv. Okay, so final thing in this project, and then I'm really finished, is we asked where do the ancient genomes fall into this tree, and I'm going to skip a lot. I'm going to skip this. This is just to show that the model that we have is consistent with another model derived with the different methods called QP-Graph. And I'm going to jump right into this analysis of ancient genomes now. So we mapped the ancient Paleo-Eskimo first. This is the ancient Sakak genome for which we have whole genome sequence data. And we asked RareCall, in which branching position do we get the best likelihood for this genome to fall onto the tree? So we estimated this position here, which is ancestral to Chukotko-Kamchatkans and Athabascans and Eskimo Aleuts here. As a second position, we tested here, where Sakak is assumed to be a sister clade to Chukotko-Kamchatkans. Next, we tested the position here, where Sakak, where the Paleo-Eskimo genome is an ancestor of the population giving rise to Athabascans, and finally another one here where it merges onto the tree after this contribution to Athabascans. And the most likely scenario is actually this one, where Sakak forms the sister clade to modern Aleuts and Yupiks right after the contribution of its ancestors to Athabascans. So this may look like a detail to you, but this is one of the things that was debated and is still debated, arguably. Okay, we also put the other genome. So this is our full picture here. The ancient Aleut, non-surprisingly, falls onto the Aleut branch. The ancient Athabascan, non-surprisingly, falls onto the Athabascan branch. And the ancient Athabascan and the ancient Sakak Paleo-Eskimo falls onto this point, as discussed previously. Okay, so let me summarize. So this is the model. I hope that you will be able to download the slides for this talk so you can look at this more carefully, because I don't have time to really go through it much. But of course, this genetic data and genetic inference doesn't really say much about where things happened. So we then took these models and tried to put them on a map and argue also with archaeological evidence where these different admixture events happened. And we actually have a model in which key admixture events happened in Alaska. So this is something you can then look up and hopefully soon read when the paper comes out. Okay, so let me sum up. Rare genetic variation is a powerful tool to assess fine-scale population structure, as we've shown both in the Anglo Saxon case and in this Native American history project. In this case, we can disentangle Athabascans, Eskimos, Paleo-Eskimos, Aleuts, and Siberian people. We have this new method, RareCall, which finally now gives us a means to actually derive a dated admixture graph, not just the topological admixture graphs from QP-Graph and other methods. And in this case, it reveals that Paleo-Eskimos are the source population giving rise to Asian admixture in Athabascans. Okay, so that's the thank you slide. My group here in Jena, of course, Eski is added in two places, here and here, because she's currently kind of between the two groups, and some colleagues that played key roles. Everyone in bold kind of played key roles in the projects I presented. Thank you very much. So I guess I now wait 5 to 10 more minutes for people having questions, which I'm very happy to answer.
Yeah, I think so.
Great. So there's a question about counting rare alleles. Is the definition rare based on modern populations? If yes, considering modern data is somewhat biased towards certain populations, is this method applicable to archaic samples with different history and locations with the same confidence? That's an excellent question. That's something that I'm kind of caring a lot about these days as well. Now, there's a simple, you can actually prove. Okay, now let me first answer the question. Yes, it is based on modern populations. So the allele frequency ascertainment, so the very definition on what we define as rare and what not, is exclusively made in modern populations. We've done some mistakes on this in the past. Actually, we've only figured this out recently, that this is the best way to do it. but you have to do it in the right way. So here we're actually ascertaining on all non-American populations. So then it works nicely, because then you're ascertaining on alleles being rare in non-Americans, and then you're analyzing American populations, ancient and modern, with their relationship to the outside populations. And then it actually works, and it's independent of any drift that happened since ancient times or modern times, because you're actually not trying to explain relationships of ancient populations among themselves, but you're trying to explain, you use these rare allele sharing statistics only to look at differences between your test samples. For example, between Iron Age and Anglo Saxons, or between Athabascans and Southern Americans. So for that, it works nicely. It's more of a problem if you start using this method where you ascertain in populations that are mixed with your samples that you actually want to study, because then you're ascertaining on an allele frequency of a related population that might indeed, as you indicate, cause biases in your test samples. So I'm sorry if that didn't make entirely sense, but I would have to revert to confer you to some papers that we're writing. This is a little bit of a tricky topic. In rare coal, I want to say, I think all the ascertainment is done everywhere. So all the populations that you put in your model, you ascertain on rare genetic variation. And then the machinery itself, the probabilistic framework, calculates all the expected allele frequencies and compares it to the actual allele frequency. So there is no problem, because you do the full spectrum anyway, the full allele frequency spectrum anyway. Does genetic have anything to do with races, is the next question. Well, I don't really know what race means, to be honest. If you mean something like dog races or races that are extremely phenotypically different, then I think it has nothing to do with it. But if you're talking about race as the concept to describe a genetic difference between one people to another people, then the first thing to say is, well, of course there are genetic differences between different peoples. And you see that, for example, in the PCA plot that I've shown multiple times. I mean, let's just look at the European one. And then you can form an opinion on what you would declare as so distinct that you would use a term like that, or whether you would use more of a Klein. Let me go all the way. I can go to the slide navigator. There we go. Okay, let's look at the PCA. Goodness. All right, here it is. So here's the PCA. And of course, you can see that there are some differences, right? People from Saudi Arabia look different from people from the Caucasus, who look different from people in Northern Europe, who look different from people in Southern Europe. But also you see that the boundaries of these populations are blurry. There's always some people who are closer to some than closer to others. So in my view, the term race doesn't help at all to describe human population diversity. Because it gives a false impression of extremely long, isolated people who have not much in common genetically, whereas the correct answer is a much more atoned one and a much more quantitative one. So I think the correct answer is human genetic differences can be quantified, and it's not so helpful to use terms that exaggerate or that suggest more genetic differences than there really are. Keep in mind, just as a final comment on this question, even between non-Africans and Africans, which is the deepest divergence in human history, you have 50,000, 60,000, 70,000 years of isolation between the two people, maybe 40,000, 50,000 more. Genetic isolation between people from Africa, some areas of Africa, than people from outside of Africa. But these 40,000 years seem much to us, but they pale in contrast to the most recent common ancestor of for example, a random gene picked from your father and mother, which can easily go back to 1 million years. Actually, the mean time of the genetic difference of the common ancestor between a gene picked from your father and your mother is about 900,000 years. or maybe 700,000, I can't remember now, but a lot longer than the 50,000 years that we have isolation from Africa. So that shows that the isolation periods that we have in human populations may seem a lot, but are not very much. We're still relatively closely related to each other. Okay, so the next question is, going back to a question asked before, numerical artifacts and unexpected peaks for the African non-African split. Could selection against Neanderthal ancestry in non-Africans have any effect on those estimates? An excellent question. I assume if the Neanderthal alleles selected against, the entire haplotypes are selected against, but if recombination is breaking the linkage, okay. Right, and so you shorten the haplotypes, and that may look the more recent. I would like to believe that, because it would give more credibility to our method, or would actually render the uncertainty of our method less pronounced. But my gut feeling is actually that plays a minor role. And why do I believe that? I think because there are so many other methods and so many other studies that have, with relatively simple methods, estimated these split times and came to a little bit older estimates. For example, the paper publishing the Alta Neanderthal sequence, I think, came to about 700,000 years divergence, with a large spreading from 500, 400,000 to all the way back to 700, 800,000, I think. And of course, these methods are based on allele frequencies, not on haplotypes. So you may have a point that selection may affect these two estimates slightly differently. But my gut feeling is anyway that plays a minor role, but I cannot really prove that. It's a very good point, actually. I should maybe think about it more. Thanks. Okay, next question. Okay, now I see that I'm talking to computational biologists. So the next question is, I just checked your GitHub to see the source code of Recall. Why did you prefer using Haskell to write Recall? Okay, if I would get a penny for every time someone asked me the question why I program in Haskell. Well, before I programmed in Haskell, I programmed in D, which is even more exotic and less used. So MSMC is coded entirely in D. So I'm not a big fan of, or let me put it like this. I love programming, and programming is, I think, more than just a tool to do science for me, but it's something that I enjoy on its own right. I read a lot of blogs and technology things about new hot ideas in the programming world and new programming languages. And I never cared much about majority rule. So just because everyone uses Python or everyone uses C++, that's not an argument for me to use C++ or Python, even though I do use them because they're sometimes useful. But Haskell is an enormously powerful programming language, a functional programming language that, if you actually get deep into it, it opens a whole new world in programming, because it's very, very different from anything else, because it's functional. And I love it because it's extremely safe. It's one of the safest languages that I've ever programmed in. Typically, if you have a conceptual bug, then the compiler will probably catch it as a programming bug in Haskell. That's not the case in Python, where you don't have any compiler, or in C++, where your compiler is very much weaker. So one of the things in many Haskellers would probably agree on is that if your Haskell code actually compiles, you have a very good chance that it's correct, and that kind of safety is a cool aspect. So I would encourage everyone who loves programming and who likes this kind of stuff to dive into that if you have the time and if you want to spend time. So if you're looking for something cool and interesting to look for to learn, then have a look at Haskell. It's a great programming language. I wouldn't want to miss it. I don't do much programming since the last 8 months or so, but I will probably continue when I have a little bit more time. But yeah, that's my answer on that.
Yeah. Do we have any other question?
So someone said bye. Good. Yeah. You're welcome. Thanks for the thanks. I don't see anything popping up.
Yeah. Could you please click Q&A?
Q&A. Oh, yeah, goodness. I haven't.
No,
okay. No, nothing there. Okay,
they use
good.
the chat box.
All right. Good. So I'm going to wait like 2 more minutes and then I think log off. Thanks for having me, everyone, so far. Okay, I think I'm going to close it then. All right, thanks everybody. Should I now click?
Just
I need
a minute, you need to
record. Stop the recording, right?
Stop recording.
That's that.